Hi. Just, doing something else. There we
Okay.
go. There we go. Hmm. AI carpentry. Don't need that.
Oh.
Okay. I'm finally back from holidays.
Oh, good.
Yes, I feel like I'm 4 weeks behind.
Yeah.
Actually, 3 weeks behind, but I've forgotten everything that I was doing before that. So the holidays really are not that great around me because it takes a week to pack for them and then 2 weeks to do them.
Yeah.
And I swear a week to unpack and try to figure out where you left off. So,
Right.
I haven't got the active matter presentation anywhere near things because I've got to do optical coherence tomography presentation for
Okay.
40 minutes.
Wow.
40 slides. I went on holidays, I had 30 slides, and now I don't know exactly what I was going to do.
Yeah.
Mm-hmm.
Well, yeah, it always gets pretty busy when you take a holiday and then it's like you come back and it's like you kind of get overwhelmed with things.
Yeah, this is why I think holidays are not always a good thing to do.
Yeah.
But I guess, well, we hadn't been able to travel to see family
Yeah.
for over 2 years or more. It was
Yeah.
more.
Yeah.
We were going to go there, and then my husband fell off the boat
Oh.
and cracked his head open.
Oh.
Yeah, so that was
Wow.
interesting.
Yeah.
He needed 10 stitches and a few other things. But,
Yeah. Oh, that sounds horrible.
well, it ruined the holiday.
Yeah.
That was about 3 years ago. So, anyway, he tried to repeat, this time without a boat.
Oh.
It worked. Boat. But this week they're all gone on the boat, so I'm going, I'll just stay home.
Yeah.
And so what's been happening since I was on holidays?
Well, I mean, we've been talking about different things. Mainak has been doing things with GSoC, and he's been presenting weekly, and he's got a pretty good handle on some of the segmentation, and he's creating an interface for it. So I was kind of hoping he'd be here this week. I don't know if he has something else, but I think he put something in the Slack, and I can't get to it right now, but and then we've been talking about other things, different papers and other types of things. So I think we've been doing some interesting things.
Can you possibly give me the references for the papers that you?
Yeah.
Sometimes they give me a lead in what I'm doing, actually.
Yeah, I have them. I don't have them right here with me, but here we go. But I'll get them together. I know we've been talking about like how to bring together resources and things like that, but yeah, I can get a list of things.
Okay, if that's, well, I know that's a lot of trouble, but
Oh no, that's fine.
you have them at least there.
Yeah, I have like the folders are organized so that I have access to everything, and
Okay, well, I really need to go online. Do you have them online?
oh yeah, you can get to them online. Actually, I can try to find. Well, I have the PDFs in the folders, but I can also give you, or I can also give you the links to them. Probably the folders would be better. I can just send you the folder links, and I can open up the permission, or the permission should be open. So.
Oh, well, I'll try that because then I could go see other things.
Yeah.
Okay. Because, yeah, I just kind of sit here and don't always look for things while I'm trying to take courses and to try to get A pluses in them is like no fun at all.
Yeah.
I only got A's, it's okay.
Yeah.
But yeah, I rarely get A's in courses. I'm more of a B student, so
Yeah.
you get a B or a B plus, and they say, "What's wrong with you? Why aren't you getting A's?" Or I don't know, because I'm me.
Yeah. Graduate school, it's not like grades are like, they're just kind of like they have to give you something. They're not really that worried about it. I mean, if you're competing for like a super high profile job, maybe, but I don't even think then. It's more like publications and things like that. So
Yeah, well, I do better with the essay-based things.
right.
That's both of these courses kind of more mostly essay and presentation, so that helped.
Yeah. So let's see. I don't know if Mainak's going to make it. I got his blog post up. We'll go over that. Actually, I wanted to look at it and just show you what he's up to.
Okay, sure.
So I'm sharing my screen. So this is his blog. He just does weekly entries for the program that he's in. And this is the work done this week, July 26th to August 1st. So he's converting the lineage population model, which is the thing that they've been using for in DevoLearn to build. Let's see if he's here. Oh, he's here. Good. I was just looking over your blog post here, Mainak. Looks pretty good. Why don't you go through it, though, and we can talk about it.
Yeah, sure. I'll just share my screen.
All right.
Okay, so can you see the screen?
Yeah, I can see it.
Here, I'll just mute myself or something here.
Yeah. Okay, so I'll go ahead and walk you guys through the update for this week for DevoLearn.
Yeah.
So last week I was actually working on making a few final touches to the GUI itself, to the GUI. Okay. So what I worked on is basically I have to convert the DevoLearn lineage population model to make sure that it works with the GUI. I have to convert it from a .pth file to a .onnx file, which was actually suggested way back in the day by Krishna. So like it's actually taken from a discussion that happened way back in the day on Slack. So so that conversion was done. This model was actually converted, and then I defined the functions that were needed to run the lineage population model via the GUI. Okay, then I had to run some tests on local hosts to actually ensure that everything works in my local environment, in the website, in the web app. Then I had to create some files using which I was able to host it online. So the files included the proc file, the setup.sh file, which is a shell script, and then also a requirements.txt file, which basically lists out the packages that need to be installed. So these files could be found in the GitHub repository that I've linked over here. Just open up the repository. Yeah. So this is the proc file, and this is the setup file that was required, and the requirements.txt file is also here. So currently I'm working on this unofficial repository, and I'll be actually moving everything that is here to the official repository very soon.
Okay.
So yeah. So the web app is actually it's actually online now. So can you see the web app?
Yeah.
Yeah. So this is the home page. It's pretty basic as of now. It just contains a couple of some of the animations that shows what the library could do. And at this point, I see this as a placeholder, and I'll be adding more content very soon to this page, to the home page very soon. So I'll go ahead and show you the models that are actually working right now online. So right now I'm actually in the hosted version of the web app. It's not a local host or anything. So I've actually placed a couple of examples here so that the user gets a quick idea of what's going on. So if the user selects one of the examples, he will get the segmentation map and the centroid map, and he could also select the other example and the input image will change, and then he will also get the segmentation map for that corresponding input and also the centroid map. And if the user has some inputs with him and if he wants to check it out, if the model works on his inputs, then he also has the option to drag and drop some files into this box, which will appear into this input part, and then we just have to press run and we'll get these outputs. So I'll go ahead to the next model, which is this one. Okay, so these are another two of the examples that I've put in this model. So this model, using this model, you can basically segment out the nucleuses from the fluorescence microscopy imagery data from C. elegans. So as you can see, there's the input that we have got to the left, and okay, I guess that's something. Okay, hold on, let me just try with another example. Okay, I think the website has crashed somehow. Let me just reload it. Okay, this has reloaded. Let's go ahead and try out some of the examples that I have. Okay, the website, I don't think. Okay, there seems to be some problem here.
Yeah.
I'm not sure what it is. The segmentation model is not really working right now online. But I'll go ahead and move on to the next model right now.
Okay.
The cell image population model, which is the one I added there in this last week. So it takes in an input image of this sort, and then it gives you a table which contains the lineage name and the predicted population of that lineage of cells. And I'm at this point I'm actually trying to get rid of this column, but it's probably to be more difficult than I thought. So this column is not really required here, this 0, 1, 2, 3, 4 column. So I'll get rid of this very soon. So these were the three models that I could integrate into the online GUI. But the nucleus segmentation model, this one, yeah.
Oh, there we go.
Okay, so this is working now. I'll go ahead and try out the other example just to make sure. I guess it's some fault with my internet, maybe it's slow, because it actually uploads the image and the output image is also pretty high resolution, and it also has to download the image.
Yeah.
So I guess that was a bandwidth issue. Okay, so the segmentation model also works. So as of now, all of the three models work, and I'm actually planning. So the screenshot that you see here, this is actually the same website, but in a separate theme. So I'm not really sure which theme to choose. I was just playing around with the theme. Like here, you can see the theme is dark, which some people might just prefer. And in this screenshot, you guys can see that the theme is light, so which some other people could actually prefer. So I'm maybe thinking of adding some sort of a toggle switch to switch between these two modes.
Yeah.
Regarding on the preference of the user. And the next goal would be to have some sort of a folder uploading mechanism or a zip uploading mechanism, using which the user would be able to upload many inputs at once and would be able to get the outputs also as a zip file, which he would be able to download. So if that could work, if that works properly, and if that works fast enough, then like it would make the job way easier than to install the library, to set everything up in the local environment and to run the code in the terminal. That could be a bit cumbersome for some people, and to have an online tool, I guess, would make the job way less easier for the scientists or the researchers who would be working on this. So okay, so I was actually, if I go back to the other blog posts, I'll just go back to some older blog posts now. Not this. I wanted to show something. Yeah, previously I was using this other library called Gradio, using which I had made some progress, but I did not decide to go ahead with it using that library because I could not find a way to host three different models in one web app using the Gradio library. And then using that library would have led to three different web links for the three models, which is not at all convenient.
Yeah.
Because here we have only a single link from which the user will be able to navigate into the other models, into into the separate models that are present. But in case of gradient, there would be 3 different links, which is not at all convenient. And
Right.
in the upcoming weeks, I'll be adding content regarding the models in the web app, specifically in the home page and the specific model pages. I'll be adding a bit of markdown text or some flowcharts which would show how the model was trained and what sort of data was it trained on, what is the architecture of the network, to give the users a better idea of what's going on. And then I'll try to render interactive plots into the online GUI, which would actually enable the users to say zoom in into the outputs and see if the model is actually working properly or not, to check the viability of the model in their data. Also, as I mentioned, I will be looking into ways of running bulk inference using the web GUI, and then I'll have to move the entire web app into the official DevoLearn repository, which I'll be doing very soon. And then moving from Travis CI to GitHub Actions has been one of my goals since the past week itself. So I'll have to get that done too as soon as possible. And then by the end of Google Summer of Code, I have plans to release a new DevoLearn version which will contain all of the upgrades and all of the new features that have been integrated into the library. And I'll also have to upgrade the DevoLearn starter notebook, which already has been done. I'm actually halfway into that. So I'll be making the change in the GitHub repository very soon. I'll be issuing a PR and which will basically merge the changes to the starter notebook. So yeah, that's it for this week. That's it for the updates for this week, and I'll just drop the link to the web GUI in the chat.
Yeah.
So everybody is able to use it.
Yeah, that's great. I had a couple questions about the blog. Could you go back to the blog post after you dropped that in the chat? So
Yeah, sure.
yeah. So let's see. You mentioned, go down a little bit. You have this predicting cell lineage populations. It outputs this table of lineages. So is it drawing from like the labeled data that you already have, or is it like, how is it connecting to the image that's uploaded?
The image that is uploaded, that's actually passed through the neural network which is the part of the backend of this web app. The neural network that is actually present in the DevoLearn repository, that's actually working in the backend of this app, of this web GUI, and the predictions are being run in real time, and the outputs that you see to the right, they're actually outputs from the neural network itself, which is present in the DevoLearn library.
Okay.
So that's how I'm getting the outputs.
So it's just making a prediction of what cells are in there, or what lineages there are.
Yeah. Yes.
So it knows this by its location, or I wonder how it's making the prediction.
Yeah, maybe the density or the locations could be like even I'm not sure what the neural network exactly looks, but like it's being trained on a large number of images. So it has actually developed some sort of a generalization, like some sort of a logic that it found out or it mapped, using which it's actually able to predict the populations to a large, like within a good accuracy level.
Right. Whereas in C. elegans, it's fairly easy to learn that because it's like the cells are deterministic. So if you're at a certain stage of development, you know kind of what cells should be there. So it's kind of like it'll know. Of course, I don't know. Do you put any sort of do you train the neural network, or does it just kind of see the image and know that it's C. elegans, or because I know you've trained it on C. elegans, but maybe not any other type of embryo.
Yeah, I've actually trained it on only C. elegans data as of now. But maybe in the future, if we get access to some more data, say some other embryo, then we might be able to expand upon this branch of this library.
Right.
So that would be done.
Yeah, and probably would have to be like species specific, so you'd have to have like a just do it
We'll
for
have
each
to have
one.
some
Yeah.
sub menus at this part, and which will basically specify the species and
Yeah. Did you have a question, Susan? Oh, you're muted. Oh. Okay, she didn't have a question. All right. Yeah, this looks...
There we go.
Okay.
There. Yay! You can hear me, right?
Yeah, I can hear you.
Oh, good. Yes. That always seems to be a problem. What language are you using to program this in? Just that base.
I'm using Python at its base, but on top of that, I'm using a library called PyTorch. So these are the two main languages that I'm using. Actually, PyTorch is a library which is built on top of Python. So
okay.
So if you ask what language, so the only answer would be just Python. That's all.
Okay. All right. Thanks. Thank you.
Yeah. PyTorch, of course, is it's well specialized for like machine learning and deep learning, and they have
I think I'm going to have to learn it because I'm supposed to be processing my images with some deep learning algorithms. Yeah, so I get to learn this.
Well, they have a lot of packages like available for general use. Like if you go, like I think, well, there's PyTorch and then there's TensorFlow, and people will argue the pros and cons of these for until they're blue in the face. But pick the one you're most comfortable with. I guess if you're doing something like a I don't know, you're probably not doing it for like a publication. Maybe you are. That might be something. Oh, you're going to publish. Okay. Well, you might want to check and see what people are doing. I don't know. What are you trying to analyze?
Optical coherence tomography images.
Okay.
So they'll just be images.
Oh, okay. Well, yeah. I mean, I was just thinking like there might be some standard toolkit for a certain type of image. Like what we have here is the we have different types of fluorescent microscopy where you have these clearly defined cells. You have nucleus markers, you have membrane markers. So this actually tends to work well for this type of thing. Then when you move into other types of images, bright field images are a different animal from this. And not species-wise, but has a different set of things that you need to do to the image. So the challenge, I think, is in pre-processing and finding maybe the right toolkit to do the work. So.
Yeah, to settle on the network and then the pre-processing part is also a bit challenging, as Bradley mentioned.
They'd be more like bright field images, I believe.
Okay. Yeah, one way to deal with that is to create masks where you take some features and you kind of highlight them and you create another data set where those parts of the cells are highlighted. You basically need something for the algorithm to hang on to. So it's like, if you have a very defined nucleus in your data, that can help you. And then, of course, if you have fluorescence markers, those are good because they really kind of, you can hang on to those pretty easily. But you're not going to have that.
No fluorescence in this. No, not at all.
Okay.
I can't find any material that is visible at this infrared range. Like there's no dyes.
Yeah.
I've asked. I've looked into it. No.
Yeah. Well, the other option is masking, where you can just take different parts of the image. Like if you have a bright field image, it's not gonna be uniform. They're gonna be you'll be able to see cell boundaries and things, and it's quite imperfect because you might take this the brightness or saturation and play with that and get maybe get it to where you can get you can do this in ImageJ where you do thresholding where you have you up the thresholding, down the thresholding, and then you have this trade-off of am I getting a lot of noise in the image or am I getting almost nothing? If you have a low threshold, you get everything with a lot of noise surrounding, say, the boundaries of the cells. If you have a really high threshold, you have almost nothing, just kind of like broken boundaries. Instead of try to find the edges, that's the key. Another way to do it is to just, and this is probably not going to work, just to do a manual sort of trace. And I think with deep learning, you can create a small training set. And I think we did this one year, where you just take some images, you trace the boundaries, and then you train the model on those boundaries, and it kind of tells you. But that's not going to be very good in some cases, because it will just really generalize. It won't generalize well. It'll pick things that look like the things you trained it on. But not every image is like that, then you might have a problem. So there's a lot of trial and error in this.
Okay. Yeah, well, I've worked with imaging before.
Yeah.
In MATLAB, but not in Python.
Yeah. Well, yeah, you should, even if you have something that's really kind of basic, you can present it to us. We have people here who are doing this, so pretty high level, so we can, yeah.
Maybe you could send me some of the data. So it would be great if I could get access to some of the data that you're talking about. Then I might just be able to get a hold of what needs to be done. So it would be great if I can get access to it.
I haven't set this up yet. It's all mostly theoretical so far. But I have taken some optical clearance tomography images of my salamander eggs. But all you can see is the blastocele because they are too soft for contrast with the infrared light. So they're a difficult image, I've been told.
Yeah.
From somebody else, another PhD trying to work with them. Yeah.
Yeah. Hello, Akshay. How are you?
Hello.
Do you have anything to share with us today, or any?
No, I've been pretty busy like the previous week. I have some summer schools also lining up. So,
Well, that's good.
yeah, I couldn't do much work. Yeah.
Oh, yeah. Thanks for joining.
Yeah, sure.
Yeah. So thanks, Mainak, for that update, and look forward to next week's update and pull requests that you have. I know you have a couple probably for me to review, so I'll review those and accept them, and looks good. So this is the first week of the, speaking of deep learning, this is the first week of the NeuroMatch deep learning course, and that's where some of our people are today. They're starting the first day of this course. So this is a course on deep learning. It's for people who are novices as well as people who have done some before. So it's largely targeting, we have the computational neuroscience course that they do, and then now they have this deep learning course, which is more specialized in terms of deep learning techniques. And I don't have the link to the site here, but I can send it out in our next email, or we'll probably talk about it next week a little bit more. But I don't know. We'll see how it goes. This is the first time they've had it, so just to let you know, those resources are out there, and I will put that in the email because I think it'll be useful for people, for reviewing methods or seeing what they have to say in their reviews, because they'll get experts in the field and they'll give lectures, and those lectures will be available online, so you can follow along as a what they call an observer track. And the observer track, it may have some useful information for people. So we'll do that next week, though. I'm going to share my screen here. So let me go over the submissions really quick. I don't think there's anything in this document that's particularly time sensitive. We still have our things we've been working on for weeks, the non-neuronal cognition, the DevoLearn papers are, on various stages of hold, some of these other projects. The mathematics of DevoWorm I'm going to talk about today a little bit in depth. And then the NeurIPS workshop. So NeurIPS, of course, is a conference that's held in December. And the NeurIPS, if you're submitting to the main track of NeurIPS, it's quite competitive. But if you are on the lookout for some of the workshops, you might be able to contribute to some of the workshops that they have. It's not as prestigious as the main conference, but you might learn some interesting things, and you may be able to get involved in this. So let me go through some of the workshops that they have accepted for this year, and we'll see maybe there's a good fit for some of our work, or you might find something really interesting. So these aren't like each of these workshops will eventually have a website. So this doesn't have the website with the submission details for submissions, but this basically is a list of workshops that have been accepted. So you might be interested in one of these workshops, and this gives you a heads up as to what's going on. So they have a lot of workshops this year. They have Bayesian deep learning. They have things on education, on new frontiers. So this is on federated learning. There's this one, medical imaging meets NeurIPS. And this is a workshop on medical imaging and microscopy imaging. I imagine there's a lot of that. There's probably a lot of medical images, like MRI images and things like that. So, that's one sort of related area. Let's see. There are things like human and machine decisions, something called Cooperative Ai. This is where they look at like models of cooperation. So I've noticed that they're getting they're going in the direction of artificial life in some cases. So some of these workshops are focusing on things like reinforcement learning, human AI interaction, game theory. So this is stuff that you might find at maybe like an artificial life conference, or even the cognitive science conference that was held recently. Then you also have some other like natural language and speech processing. I'm not sure if there are any other ones that really are relevant to this group. There's deep reinforcement learning, so if you're into reinforcement learning, that might be interesting to attend or find out more about. AI for Science, Mind the Gaps. This is a workshop where they have it's sort of like computational biology, chemistry, material science. So they're looking at these critical gaps that exist with sort of applying algorithms to science. And so, despite this promise, several critical gaps stifle algorithmic and scientific innovation in AI for science. Unrealistic methodological assumptions or directions, overlooked scientific questions, limited exploration at the intersections of multiple disciplines, the science of science, and responsible use and development of AI for science. So they kind of go over these gaps. That might be something interesting to review and be on the lookout for. I don't know too much about what will happen there, but you have things like data-centric AI. You have meta-learning. That's always an interesting topic. Offline reinforcement learning. Like things like the symbiosis of deep learning and differential equations. What else? Just a lot of different topics, kind of exploring some niche issues in the field. So ImageNet, past, present, and future. So this is since its release in 2010, ImageNet has played an instrumental role in the development of deep learning architectures for computer vision. So a lot of the microscopy images are sort of, relies on this computer vision model of, analyzing images using feature space. So this is sort of a focus on the ImageNet architecture and kind of a reflection on that. So there are a lot of different things you can look at. I would look at that link and see what's there. It kind of gives you an idea of the state of the field. The other thing I'd like to talk about is this mathematics of DevoWorm. So this is this poster we're working on with different equations and different models that we might use in that sort of we hit on over week after week in our group. So we have tools like the von Neumann neighborhood, which are essential for understanding cellular automata. We have these models of the embryo and these like multi-parameter models. We have things like neural networks and complex networks and lineage trees, which are all different versions of, with something that has nodes and links between them. So, we have neural networks, of course. They're very different from complex networks and lineage trees, but they all are used to represent this developmental function at some level. And then we have some other newer ones that I put in here just last week. So last week I talked about creating a differentiation tree with a computational agent. And so this is an example here of this agent that's being developed in, actually that should be there, but this is an example of an agent that's being developed from like this 3-layered embryo, and it's being developed into what they call a Bratenberg vehicle. And so this is a differentiation tree of that process. We can also use what we call developmental game theory to look at divisions in embryos, divisions of cells. And so that gives us some maybe some information about why cells divide and move into different parts of the embryo beyond like the typical model of biophysics and things like that. And then I'm also interested in putting in a category theory section, and this is something we talked about in one of the papers that we just published in the last 6 months. And so I want to go back to that and sort of put a visual model together for this. And then eventually, again, like I said, we want to maybe create a preprint or a little chapter on this where we take each one of these models, and I have them in circles because this is sort of showing how they're related to one another. Maybe I'll do away with that, but to get each one of these little circles, whatever's in them, and give them their own little section, and then write up a little bit of information about it. So this is coming along. So that's all we have for that. Thank you for attending Mainak. So next thing I want to do is go into the papers. I don't think we need to review anything on the GitHub repository here. We have our major tasks. We updated this recently. So let me go to the papers. So I know Susan was asking about the papers before, sort of at the beginning of the meeting, and I told her that we're keeping things in these folders and they're open. I don't send the folder links out as much as I should, but to let you know that we have these folders, and I sort of been organizing them a little bit better with different subtopics. So just to let you know that, and this is an example of the subtopic structure here. So one of the things we've been doing is talking about what is environment. So this is the 4th installment of this, and so this goes back to a question that I had, or a conversation I had with Dick, and I've talked about this before, about environment. And he asked, what is it, what is its relevance, and how do you define it? And it was sort of a rhetorical question, because we kind of know what it is, but we kind of don't know what it is. There is a lot to sort of quantify and really define. So, we've been talking about these points that environment is permissive. It allows things to be transduced into the biological system. It allows change to happen in development. And environment is also instructive. So when this information is transduced into the system, it allows for change in development, but it also provides specific information to the system. So that's so this week I'm going to talk about something called immediate early genes and activity dependence. So the first paper here is this paper, Synapse Development Organized by Neuronal Activity Regulated Immediate Early Genes. So what are immediate early genes? And they talk about this. So classical studies have shown that neuronal intermediate early genes, or IEGs, play important roles in synaptic processes critical for key brain functions. So these IEGs are genes that are sort of the first line of response to something in some environmental stimulus. So IEGs are transiently activated and rapidly upregulated in discrete neurons in response to a wide variety of cellular stimuli. So, this could be actually stimulating a single cell. It could be stimulating a nervous system where you get like a sensory input. And so and sometimes it's also with respect to stress. If the organism experiences some stressor, it can activate these immediate early genes. In this case, they're actually uniquely involved in various aspects of synapse development. And of course, we know that synapses are notoriously, or maybe maybe not surprisingly plastic, that they change. Sometimes they are maintained in development. Sometimes they change quite a bit in development. And so this is a good place to look at the effects of these immediate early genes. In this review, we summarize recent studies of a subset of neuronal IEGs in regulating synapse formation, transmission, and plasticity. We also discuss how the dysregulation, or how these things are not regulated in the same way, of neuronal IEGs is associated with the onset of various brain disorders and pinpoint key outstanding questions that should be addressed in the field. So they talk about some of these pathways here in different model organisms. So classic immediate early genes are FOS and Jun. So these are genes that are upregulated maybe by some stressor or some sensory input. They've also identified a number of transcription factors that are related to the expression of these genes. So these are things when the gene is when it's transcribed, it leaves these transcription factors in the cell, and this is something that drives other processes like protein protein construction and other things that could regulate other genes. They're very diverse, but basically if you find transcription factors of these genes, it's being expressed. So this kind of goes through a lot of this. This is a synapse, and this is sort of the target here of activity-dependent transcription. It produces these transcripts, which are mRNA, and it leads down these paths to the synapse to regulate what's going on at the synapse. So the other paper is this Emerging Themes in Neuronal Activity-Dependent Gene Expression. So this is a review article. This is a little bit different. It zooms out from the synapse, and it kind of talks more generally about these. So in this review, we attempt to discuss emerging themes in the regulation of neuronal activity-regulated genes, focusing primarily on an important subset of immediate early genes. Okay, we first discuss earlier studies that have illuminated the role of cis-acting elements, which are elements that are near the gene itself that regulate the gene within the promoters of immediate early genes. So these are elements that are within the promoter region. And, if you're not familiar with gene expression, that's fine. Just let it know that this is one version of of types of elements you can have. The corresponding transcription factors bind these elements, and the roles of major activity-related signaling pathways. Then they talk about how this has an important role for epigenetic and what they call topological mechanisms. So this is where you have both sort of regulating how genes are expressed, and then these things where the RNAs are folding and taking on different shapes, and it actually affects that, which in turn then affects how proteins are built, how the RNAs are translated, and things like that. So then the third paper then is this role of immediate early genes in synaptic plasticity and neuronal assemblies. So this is where it actually affects the memory trace. So this is where it's affecting memory of the organism. And This is, again, they talk about the immediate early genes, EGR1, CFOS, and ARC, and it's rapidly and selectively upregulated in subsets of neurons or specific brain regions associated with learning and memory formation. So now we're talking about learning and memory formation, but we're talking about the molecular level. So these different IEGs have been widely used as a molecular marker for neurons that undergo plastic changes. So when they're looking at long-term memory, one of the things they can do, say in a mouse model, is look at the expression of these genes, and it tells them something about what may happen in terms of memory formation. So they've been able to study these genes using different types of studies, different types of measurements, and it's revealed that during learning, IEG positive neurons encode and store information that is required for memory recall, suggesting that they may be involved in the formation of a memory trace. But we know that a lot of these processes remain unclear. The only thing we really know is that these genes are involved in sort of setting up a lot of the response that goes into sort of the plasticity of neurons and then eventually to learning and memory. So that's that part of the environment. So in this environment theme, we've gone from looking at stimulus to looking at what happens within the cells, within the nervous system, and this tells you something about environment, how it's affecting both the organism development and more generally. So I hope that, if you go back to the YouTube videos, you'll see that I hope we can stitch that together a little bit. I'm going to go back and look at them and see if we can come up with some sort of summary statement on what we're talking about here, and what are the major themes. I think that's important, though, to talk about at different levels of organization. So from the organismal level, where you have sensory inputs, to this molecular level, and the molecular level may seem unclear as to its relevance, but a lot of studies in neuroscience and in developmental biology, they often use molecular markers to mark out some of the the hallmarks of these processes. And so this is something, again, if we're looking at microscopy images, we're going to see these. We can actually do some really interesting things with some of these markers, because they'll often use a fluorescent marker to mark some of the expression of some of these genes, and you can generate or work from a data set where you have these things put together in time, so you can actually look at the dynamics. So it's really, there's some interesting stuff that might happen in the future on that. So this one is interesting. This is early embryos and emergent embryos. So this is a collection of papers here where we have a number of different model organisms, and it's sort of about what's going on in the early embryo and things that are emerging. So there are different things like the emergence of an embryo itself, the emergence of behavior. So the first one, well, actually, no, I'll start here. This one here, because this one starts off in time as the earliest one that we have. Early Ediacaran cavius fiara, if this will open up. All right.
Try downloading it and then opening it if it doesn't look that way.
Okay. Oh, hi, Krishna. How are you?
Hi, I'm good. How are you?
All right.
Just came from the Neuro match class.
Oh, how was it?
It was great. Great experience. It was hectic, but really loved to,
Yeah.
It was great experience.
Well, that's good. I'm glad you enjoyed it. We were going to go over it maybe next week, a little bit about
Sure.
All right, let me start over here with this. Okay, here we go. So this is actually one of the first embryos that were found. We talked about this before. We talked about one of the first embryos that were found. This is, I think, a different embryo. This is a different assemblage where they kind of talk about the first embryos in plants. This is now the first embryo, I believe, just kind of showing some of these processes that you see in modern embryos. But this is 609 million years old. So this is very, very early. This is before Cambrian explosion where you get a lot of multicellularity. So this is like very early multicellularity. This is so Cavus Viara was an enigmatic component of the 609 million year old wing gum biota of South China. This study uses an X-ray tomography to characterize cellular structure and development. So they're taking these from fossils and they're actually looking at some of the things that have been preserved. So first of all, you have this thing that's being preserved. I don't know if we want to call it an embryo, but it's definitely some sort of collection of cells that exhibit some of these like embryonic features. So you have these things like this organism develops within an envelope by cell division, ingression, detachment, and polar aggregation in a manner analogous to gastrulation. Now, it's not gastrulation as we know it today, maybe, but it has this sort of set of features. Together with evidence of functional cell adhesion and development within an envelope, this is suggestive of a holozoan affinity. So this is suggestive that it's similar to our development today. Animal development and plant development, I guess, too. It has. So this is the This is in this Dalshanto formation. So this is again. We go back to this. We've talked about this in weeks past, where they have these. It's a rich microfossil assemblage that preserves biological structure to a subcellular level of fidelity and encompasses a range of developmental stages. However, the animal embryo interpretation of the main components of the biota has been the subject of controversy. Here we describe the development of Cavus Viara, which varies in morphology from lensoid to hollow spheroid cage. So this is the cage up here, and they have these different features of it. So they've been able to pull a number of different examples from this biota. And, there's this controversy about whether these are really kind of like early embryos or not. But they do have these features that look like embryos. So the idea is that in this Ediacaran period, which is around this time of Earth history, during which molecular clocks estimate the fundamental animal lineages to have diverged. So they're using like what they call a mutational clock to calculate back to a common ancestor. And the calculations suggest that this is about the time that this happened. So this is evidence for this maybe being a common ancestor to our embryos that we have today.
I have a question.
Yeah.
Sorry to interrupt.
Yeah.
It's like something like similar to tracing the phylogenetic trees.
Yeah, it is. That's exactly what they're doing. They're using molecular sequences, and instead of figuring out the common ancestors intermediate to this earliest common ancestor, they're looking at the mutations, and then they're estimating like a mutation rate for each mutation, and then they can just work backwards and say, this is the common ancestor. Given the number of mutations we see, this is how far back it is.
Okay.
Yeah, so it's a method that's imperfect, but they use it to. It's actually, depending on the data that you have, it can be very precise.
Mm-hmm.
So these cage-like and solid morphs have been interpreted to reflect intraspecific variation, but instead represent members of a more continuous spectrum of morphological variation. So that means that there's this we don't really know. There's some variation. We don't really know if it's within the same species or whether it's like there's a continuum of variation or whether they represent different species. So this is, it's pretty unclear as to kind of, in paleontology in general, you can't really get good species estimates. They're always fighting over it. And sometimes, if you find different parts of the same organism, you can't say for sure whether it was part of one organism or multiple organisms. So it's never really clear. Nevertheless, here's some nice microscopy images of these structures. So these are the structures here they're talking about. And they're looking at some of the formations that they make within the shell. So this is sort of like the embryo where you have an eggshell, and then you have these cells inside that are moving around and twisting and forming patterns. And then this is the different developmental stages. So they go through developmental stages, or what they assume to be developmental stages. And then they have this developmental biology and phylogenetic affinity. So this is kind of like Krishna's question about like what they look like and where they fit into this tree of life. So they fit in down here at the bottom, and you have all these different descendant species or descendant groups. So we're in Eumetazoa here. And then there's Porifera, which are sponges. There's Choanoflagellata, which are like flagellates, which have like these little arms and marine organisms. And you have spores and other things here. So you can see it just covers this vast distance across the tree of life. And they all have this sort of common ancestor, which is basically this thing with a shell with some cells inside. And they have lipids and they have cell mass. And they make these things, there's an experimental model that they use that are like these little lipid spheres that they can make. And they actually use these to look at different aspects of pattern formation and of morphogenesis. And I can't remember too much about them right now, but they use them actually in biomedicine to look at like, they're sort of looking at like how cells form and embryos form. So this is an interesting parallel, and I'll try to get a paper on this thing I'm talking about next week. But yeah, this is kind of reminiscent of that. It's basically a very basic model of a cell or of an embryo, or actually a multicellular structure. Yeah. So yeah, this is a nice paper to look at for that sort of perspective. Another thing we have here is booting up the organism during development, pre-behavioral functions of the vertebrate brain and guiding body morphogenesis. So this goes now to frogs or Xenopus embryos. So now we're in the modern era, and we're looking at these Xenopus laevis embryos, which is a common model organism that shows that the very early brain has important functions long before behavior. And we know that from our group. We know that there are these different movements that the cells make, and they're doing things to engage in pattern formation. This is before, the brain sort of starts to form, and then behavior comes after that. While the nascent brain is being constructed, it is required for normal patterning of the muscle and peripheral nerve networks, including those far away from the head. In addition to providing important developmental signals to remote tissues in normal embryogenesis, its presence is also able to render harmless exposure to specific chemicals that normally act as teratogens. So this is like things that would kill the embryo or some developing tissues before they get a start. They can, monitor the embryo for these things. These activities of the early brain can be partially compensated for in a brainless embryo by experimental modulation of neurotransmitter and ion channel signaling. So this is Michael Levin's group, and of course they do this stuff with bioelectricity, and so that's one of their interests. And so you can do things really early to the embryo really early on, and all embryo cells have this sort of bioelectric potential. So this bioelectric potential is acting to regulate the embryo well before it's generating any sort of behaviors. Here we discuss the major finding of this paper in the broader context of developmental physiology, neuroscience, and biomedicine. The novel function of the embryonic brain has significant implications, especially for understanding developmental toxicology in the context of pharmaceutical reagents. So they're just really interested here in looking at what the cells of the embryo, maybe the emerging brain, does before it starts to form synapses with the muscle and starts driving muscle activity. So what will happen is you'll get this emerging nervous system, you get these neurons that emerge, you start to get connections, you get these what they call gap junction connections, and then this brain starts to form. Later you'll get synaptic connections, and then you'll get some electrical activity in a network. Now in parallel you have the muscles forming, and the muscles will start to twitch sort of autonomously before they're wired to the brain. So these axons will go out to the periphery, make a connection with the muscle, and once that happens, then the muscle is actually controlled by the brain. But before any of that, then, you have this system where they're actually, the cells and even the brain is looking for things that might hurt the embryo, and it's regulating those things. And so the brain is serving a function even before it starts connecting to the muscles. So this is really an interesting piece of work because it suggests that there's this very early brain is actually doing more to regulate morphogenesis and patterning of tissues than it is driving behavior. And so it's actually interacting with its periphery, which is something that happens when the connections come into play and it's connecting to things like muscle and it's driving behavior. It can also regulate that periphery earlier. And so that's kind of the point of this paper. And I'm not going to get too much more deeply into that, but I did want to talk about two other papers here. This mouse embryonic stem cell paper. So this is where you have these mouse embryonic stem cells that self-organize into trunk-like structures with neural tube and somites. And this is a paper on this. I'll just look at this figure here, where you have this trunk-like structure. So what they're doing here is they're forming these embryoids, and we've talked about embryoids and different structures like this before, where you have these three-dimensional stem cell culture systems, where you have a bunch of embryonic stem cells. You allow them to aggregate into a gastroloid, and then you embed them into this structure, which is a trunk-like structure. And then you can observe their behavior when they're put onto this three-dimensional structure. They're doing a lot of things to form some sort of shape. And so you can actually do things when they're on this scaffold. You can expose them to different chemicals. And these chemicals are things, genes that are expressed in the cells, but you're giving them more of it. You're modulating using these chemicals. You're getting, you can look at like single cell RNA-seq. So you can actually take the cells, each cell, you can sample what their transcriptional profile is, and you can look at this over time so you can see what each cell is doing to its neighbors. So we talk about cellular automata. This is actually a cellular automata where we can reach in and see what the metabolic activity is or what the gene expression activity is. And this is in a context of some geometry. So we know that they're interacting with their neighbors and it's influencing what they're expressing. Maybe it's expressing genes that allow it to differentiate into a certain type of cell other than a stem cell. Maybe it's suppressing its potential. We don't really know, but we can look at that using this method. So we have this TLS, which is the trunk-like structure, and it's supposed to resemble an embryo. So we can map between this TLS and an embryo, and we can look at what's going on with respect to the 3D structure of an embryo. Finally, we can do genetic manipulation, so you can actually manipulate cells genetically and then put them in this context, and they may have different behaviors when you knock genes out. And so this is all kind of goes through this paper. So they're just basically creating this matrigel scaffold. This allows us to build this trunk-like morphogenesis from these cells. You build this scaffold, you embed the cells in it on the surface, and it will form these complex structures. So this is a picture of the gastroloid, and this is the trunk-like structure, which resembles, so this gastroloid is like not shaped by a scaffold. This is just a gastroloid that's grown spontaneously, and it's allowed to grow up to this structure. And they're showing here that the trunk-like structure is similar in a lot of ways, and you can grow them in different ways. Then they show this in comparison to the E9.5 embryo, and this is like a certain stage of morphogenesis in the embryo, in a in situ embryo. And they have other images here. So generation of trunk-like structures with somites in a neural tube. So you can actually take these structures and form things like somites in a neural tube along them. So you can actually direct the differentiation of cells in certain ways using gastroloids. Okay, so that's that paper. And finally, we have this paper on axial elongation of caudalized human organoids. So this is another organoid paper, but it's focused on neural tube development. And so the abstract here is the axial elongation of the neural tube is crucial during mammalian embryogenesis for anterior posterior body axis establishments. This is head to tail axial organization and subsequent spinal cord development. So we need to have this early on in order to have a functional embryo. But these processes cannot be interrogated directly in humans as they occur post-implantation. Here we report an organoid model of neural tube extension derived from, again, human pluripotent stem cells. So this is a stem cell that you make from differentiated cells, but they behave like stem cells. These aggregates are what they say caudalized with Wnt agonism, enabling them to recapitulate aspects of morphological and temporal gene expression. So this is just a construct they're putting into the cell, and they're getting, or into the structure, so that there are these signals that they use. One of them is Wnt signaling, and it's supposed to shape it gives it an orientation. So they basically, this Wnt agonism acts to orient the cells in one direction or another. And they're able to caudalize it, meaning they put it towards the tail end of the structure, and they're able to get the expression that they desire. So they're able to recapitulate a lot of the morphogenesis and temporal gene expression patterns of neural tube development. So they're able to replicate it.
Did I say something?
Yeah.
Some of this is physics, of course.
Yeah.
And there's, what's it called? Called Raleigh segmentation. I don't know. Anyway, if you lay a string of honey out on a piece of wax paper, it will break into segments. And so that's physical force, and part of this is physical force. And they haven't got it incorporated into their science yet.
Huh.
Anyways.
Yeah, I didn't know that. I didn't know that. Well, I've heard of some of these things where, it's actually like, you have physics that act in the same way, where there's like this physical effect that, a lot of these experiments, they're using a lot of these signaling mechanisms, but when they take the cells out of the embryo context, it's interesting, but you're saying that there's like a physical thing that they should be investigating in these models as well.
Yes.
Yeah.
Yeah, it's the two things, the molecular and physical. And they need to add the physical to this. Anyway, okay, bye. Krishna's leaving. So.
Oh, okay. Have a good day, Krishna. Okay, bye.
So.
Bye.
Bye.
You too. Have a good week. So this is this basically goes through the molecular aspects of this. Critical threshold of Wnt agonism stimulated single axial extensions while maintaining multiple cell lineages. The organoids displayed regionalized anterior to posterior Hox gene expression with hindbrain regions spatially distinct from brachial and thoracic regions. So we talked about Hox genes before, how you have these different genes that regulate different parts of the segments of the embryo, and these correspond to these different gene identities. And so they do some CRISPR, which is to modify the genome, modify how things are expressed. And so together these results demonstrate the potent capacity of caudalized PSC organoids to undergo axial elongation. So I think that, and then that has relevance to nervous system development. So I think that group of papers maybe was a little bit spread out, but I think there's definitely a theme there, and I wanted to go through that. I wanted to kind of tie together some of the stuff we've been doing with like ancient embryos and kind of modern embryos and things like that, and behavior as well. So let's see, we have some things in the chat. Oh, you wanted to say something, Susan?
Oh, it's just, I wondered what the titles of the last two papers were, because I actually had somebody come to my door and say the cows were out, so that's
Great.
over everything.
So let me see. I'll share this with you, the folders. These last two, this is the axial elongation of caudalized human organoids mimics aspects of neural tube development.
Yeah, that one. Axial elongation of... Whoops.
I'll put it in the chat here.
Oh, okay. I can just copy and paste them.
Yeah.
When things are in the chat, I copy and paste them into a Word document that I've already, it's called DevoWorm 29.
Okay. Yeah.
Why should we do that?
Let me put the other in the chat too. This is the mouse embryonic stem cells self-organizing in trunk-like structures with neural tube and somites. This is the second article.
Okay. Yes, I'm actually trying to see if I could do some measurements on this rally. It's rally instability.
Oh yeah. That's the physicist. Yeah, or the
It's an instability of a long tube of viscous fluid.
Yeah, I've heard of that, and so that's something that we haven't. We sometimes we talk about the physics, sometimes we talk about the chemistry, and it's kind of interesting how they intersect. And there's also, of course, soft active materials, which is, I think, something you're probably into as well. And they've been able to take soft active materials, things like gels and sand and things like that. And they've been able to actually observe a lot of things that look like they're sort of like biological pattern formation. So, and even in like things like colonies of rod bacteria, they can observe the same types of things where they behave sort of at the collective level as this active material that has different features to it.
Okay, no, I haven't. Do I have that one? I don't have the paper on that, but yeah, I'm interested in looking at that, because then you can kind of maybe direct just an experiment.
Yeah.
In that way, even if it's just some gel that you're observing with your optical coherence tomography.
Right.
And to try to discern the pressures that are on each segment or something. Like how does this change? So even if you did that, it would be a unique experiment that nobody's done.
Yeah, if we go back even to like the early embryos, and we don't really know what early Earth history was a bit different than now. You had like ice ball Earth, snowball Earth, and other like environmental things going on at the time. So it really might be interesting to see like these early embryos. What kinds of physical forces were they experiencing? Were they very similar to what we have now, or because embryos sort of evolved in this, or we can see maybe they already kind of had a shell, but why do we have eggs?
Mammalian embryos have to have high CO2 content and high temperature.
Yeah.
Otherwise, they don't develop.
Yeah. And that's of course, Earth history, you had a lot of periods where you had fluctuations in CO2 and temperature, and it's like we didn't, of course, start with mammalian embryos, but we ended up with, they had to have similar conditions in the cell. It's like we had eggs, and then we had embryo. We would move to like mammalian embryos. You'd have that as like an option, but what were the conditions that they needed to have?
Yeah, it's interesting. Well, embryos run the whole gamut of Earth conditions. I mean, the bacteria that grow in very hot or very cold temperatures, so we can go from there, I guess.
Yeah. Yeah. Okay, well, I think that's it for today. I think I'll sign off, and next week I'll send you those folders that we have.
Okay. Because I feel like I've missed something.
Yeah, well, we go through different things, and it's always hard to like keep up with these unless you go back to the YouTube video. And so I'll go and I'll get some of the materials and send them off to you.
Okay, thank you.
Yeah, and good luck with your things for your graduate studies with the presentation you have to make.
Yes.
Yeah.
It needs to be good. We'll just put it that way.
Yeah. Yeah.
I've probably irritated my professor enough to
Oh,
last.
yeah.
Oh well.
Yeah.
Thanks, and we'll see you next weekend.
Yeah.